Good morning, everybody. I am Nobuo Shimamoto, an emeritus professor of National Institute of Genetics. This is Mount Fuji in Japan, near my institute. It is about 3,000 meters high, but it is not too difficult to go to the top in summer because of the public transportation to the middle. And thus, it attracts a lot of people. In this webinar, I am telling that some proteins are more attracted to their specific sites on DNA, like Mount Fuji top, by one-dimensional diffusion along longer DNA, not only in the speed, but also the affinity. The effect on the affinity has been disregarded as a violation of the thermodynamic rule for decades. But I show here it is actually not. Measurement of KD of triple binding. Triple protein and triple site on DNA are our materials. A famous protein equally triple R, a pair of gray drops here, is a repressor of TRIP and other operons, and has a specific binding site called triple O with a co-repressor L-tryptophan. It is a homodynamic molecule composed of two identical subunits, namely a typical bacterial repressor. We use triple site of the repressor gene TRIPR. It is 18 base pair long with this sequence. We cloned it at the centers of various DNA fragments from 18 to 4,000 base pair. In determining the affinity, we use hydroxyl radical footprinting of DNA, one of the most reliable methods to measure a site-specific protein DNA binding, because the signal is strictly linear to the amount of the complex. The DNA is cut preferentially at the position where triple is bound. Then we extend the footprinted DNA with a P-specifically labeled primer DNA and express it. This is the autoradiogram. Please note that triple binding does not protect DNA because hydroxyl radical is extremely small, but instead the binding enhances the cutting probability because of strong local electric field in the complex. The specific cut shown by the arrow was observed only at triple position, and it disappeared in the absence of co-repressor tryptophan, as shown in the control lane denoted -TRT. These band densities of this autoradiogram were then quantitated. The band densities are plotted against triple concentration. It shows a hyperbola as shown as the solid line in this figure. This hyperbolic curve is consistent to the one-to-one binding as shown in this equation of KD. The KD value for each DNA was determined by least square fitting to this equation. These experiments were made by Takashi Kinebuchi. This is the experiment to obtain the dissociation equivalent constant KD. The result was surprising. The observed affinity changed 10,000-fold according to the DNA length. The DNA length dependence of binding affinity is named antenna effect, irrespective of the mechanism. This is the largest antenna effect so far reported. KD value, the inverse of the affinity, rapidly decreases up to several hundred base pair and levels off for DNA lengths longer than kilobases. There is only one conventional mechanism explaining the DNA length dependence, namely an antenna effect. The mechanism is schematically explained here. The one-to-one complex of triple and triple site shown at the left of the figure, forms the second contact with another DNA site, forming an intracellular DNA loop, as shown in the right. This loop complex is supposed to be more stable due to the second interaction. This mechanism is actually proved for enhancers and repressors like ratchet protein. The longer the DNA, the more probable for forming the DNA loop because of the rigidity of the double-stranded DNA. Very reasonable. However, this mechanism was denied by the following experiment. Looping does not cause the antenna effect of 2-power. We connected 36 base pair triple DNA with a 214 base pair non-specific DNA in two ways. One is by regular phosphodiester bonds, and the other is by biotin avidin that has been proved to allow the looping mechanism in the case of an enhancer. From the top line to the bottom of this table, we examine the affinities of 36 base pair short DNA, 250 base pair DNA, 250 base pair DNA with avidin-biotin connection, and short DNA with avidin at its end. The phosphodiester connection gave us 7.4-fold antenna effect as shown in the relative affinity column. However, biotin-avidin connection perfectly removed the antenna effect and showed the affinity almost the same as that of the short DNA. As a control, avidin addition at the end retained the affinity the same. Therefore, the 7-fold antenna effect is caused by some movements that are blocked by biotin-avidin bound at the distance from trip O. A likely movement is, of course, one-dimensional diffusion of tripper protein along DNA. Contradiction with detailed balance. This result is very puzzling and contradicts to our conventional understanding of chemistry. We measure the affinity surely at a stationary state because the affinity did not change during the incubation period from 5 minutes to 2 hours, long enough for a biochemical assay. There are two pathways, direct and indirect, as shown in the left and right in this panel. The indirect pathway is mediated by one-dimensional diffusion along the non-specific DNA, and it may accelerate the pore association and dissociation of the binding. However, the free energy of the complex is determined only by the interactions between 2-power and 2-pole, and thus should be independent of DNA length, namely the binding equilibrium, and thus the value of KD or affinity should be constant. This rule is called detailed balance, a thermodynamic rule. The rule has been proved by statistical mechanics, and it is almost tautology of microscopic equilibrium. We got stuck at this stage for decades. Thus, I formed a small meeting with 3 theoretical people as well as 2 experimentalists. The theorists examined all possible interpretations, and the experimentalists examined possible artifacts in our experiments. It lasted more than a decade, and the oldest member, my mentor Junichi, passed away during the discussion period. But in 2019, we reached the final conclusion. In short, our conclusion is that the binding converges to a macroscopic stationary state containing microscopic non-equilibrium. Stationary state with microscopic non-equilibrium may sound something new to most of the audience. So as me, we had been brainwashed in our chemistry class. Most regular reactions were supposed to converge to an equilibrium both macroscopically and microscopically. Macroscopic equilibrium is observable and can be experimentally confirmed, as we did for TIPA. But there are no logical requirements for microscopic one in converging state. It is merely an assumption, and theoretical peoples have already started discussing stationary state with microscopic non-equilibrium. Unexpectedly, triple system may be the first real and familiar example of the macroscopic stationary state with microscopic non-equilibrium. Let me explain a little bit more. A spontaneous conformational change, as low as or slower than the binding, can shift the binding equilibrium, including a reaction flow towards a new equilibrium. Such a microscopic flow occurs randomly, but the direction is always towards a new equilibrium. It results in a microscopic circulation and macroscopic circulation flow among reaction states. No violation of physics because detailed balance does not hold at microscopic non-equilibrium. Mikito named this mechanism chemical ratchet after the terminology by Feynman, a good name. In the case of triple binding, the chemical ratchet is explained as follows. The upper line is the kinetic scheme for the binding mediated by one-dimensional diffusion, and the lower line is the kinetic scheme of the direct pathway without one-dimensional diffusion. Let me start from the equilibrium of the upper. By rare thermal fluctuation, the complex A is distorted to be the unstable complex B at the moment. In other words, the potential of complex thermally fluctuates at non-equilibrium. Then the complex B is dissociated to attain new equilibrium and followed by the formation of the stable complex A again because of the original equilibrium is in favor of the complex A. This is one cycle. Therefore, the upper is the major association pathway and the lower is the major dissociation pathway. The switching of two kinetic schema is as slow as or slower than the binding reaction, but before the measurement of the affinity, the system turns over many cycles to be stationary. It is noted that a circulating flow is generated in macroscopic stationary state. Again, it is not a violation of detailed balance because this rule does not hold in the presence of microscopic non-equilibrium, which is alternate switching. Then, Shigetoshi constructed a general equation containing one-dimensional diffusion by combination of a diffusion equation with rate equations. The resultant composite equation can be solved analytically under a stationary state. With a stationary state solution, Kd is expressed by a hypertangent function shown here. The blue line is the best fitted theoretical curve and gave 18 base pair for the size of tripole and 635 base pair for the diffusion distance of one-dimensional diffusion. They are pretty reasonable values. Alternatively, I solved the composite equation supposing that the one-dimensional diffusion is more rapid than the binding to DNA. Such condition is realized for DNA shorter than diffusion distance, and the non-specific complex distribute uniformly over DNA in that case to give a set of solvable rate equations. The resultant Kd is shown in red. And again, it gave the same size of the triple site 18 base pair. I here show one possible molecular model that the slow conformational change is the DNA bending at triple position, although there are many other possibilities. Let me talk about the biosignificance of chemical ratchet caused by one-dimensional diffusion. The diffusion is known to accelerate repressor binding to its operator. However, the typical time scale of producing mature RNA is a minute in E. coli, but the time scale of repressor operator binding is typically only a second or less. Therefore, the production can become faster by only a second or less. This cannot be a radiation. In kinetic radiation, the time scale matching is essential as explained in this reference. In contrast, increasing the affinity of repressor operator binding by chemical ratchet can decrease the amount of active initiation complex by orders of magnitude. Then, initiation can become slower by orders of magnitude to be rate limiting in the RNA production. Therefore, one-dimensional diffusion enables a repressor to inhibit transcription through chemical ratchet mechanism. Chemical ratchet makes one-dimensional diffusion to regulate transcription. Our novel proposal is the crosstalk at a distance. We constructed a low-copy plasmid expressing LAC-Z marker gene. We determined the triple repression ratio by measuring LAC-Z activities with and without the co-repressor L-tryptophan in the culture medium. In the plasmid, we introduced the binding site of another protein, LEX-A, at the site 79 to 98 base upstream of the triple promoter. From this promoter, LAC-Z is transcribed. We transform E. coli with this plasmid so that the endogenous LEX-A can bind to the binding site. The bound LEX-A is difficult to contact to triple and RNA polymerase because of the long distance, but can block the one-dimensional diffusion of triple. Therefore, when LEX-A is not bound, triple can freely diffuse into triple with affinity increased by chemical ratchet, leading to high repression ratio. When LEX-A is bound, it partially blocks the one-dimensional diffusion triple, decreasing the affinity for triple, and the repression ratio should be decreased too. The results of the in vivo experiment were consistent to this prediction. We prepared 3 plasmids having no LEX-A site, low affinity LEX-A site, and high affinity LEX-A site. In the absence of LEX-A binding, the ratio was about 4, and with weak and strong LEX-A binding, the ratio decreased to 1 as expected. With high affinity LEX-A site, the repression by triple was perfectly suppressed. When we overproduce triple, the ratio is more sensitively responded to LEX-A binding. The results are independent of the distance between 2 sites, indicate the absence of artifact caused by the direct contact of LEX-A with the transcriptional machinery. Therefore, crosstalk at a distance can exist. There are 5 conclusions as shown here. Number 1, a reaction can converge to a stationary state with microscopic non-equilibrium. Number 2, in such case, a circulating flow among the reaction states can exist because of microscopic non-equilibrium. We name the mechanism chemical ratchet. Number 3, the affinity of a DNA binding protein for its specific site can be enhanced by its one-dimensional diffusion. Number 4, a rapid protein DNA binding can regulate much slower gene expression by an antenna effect caused by one-dimensional diffusion. Generally, the kinetic regulatory mechanism must have the time scale similar or slower to that of the process to be regulated. A binding process is usually much faster than the gene expression and thus changes in the speed of the binding reaction cannot significantly control gene expression. You require slow conformational change or covalent modifications to make the timescale to match. But if regulating process changes the binding affinity by chemical ratchet, the timescale matching is not required. Number 5, two different DNA binding proteins can crosstalk by inhibiting the one-dimensional diffusion of the other. If a regulatory protein shows an antenna effect due to one-dimensional diffusion, the diffusion will be inhibited by the binding of another protein to a site near but at a distance, leading to the crosstalk of the two proteins. Thank you for your watching my presentation. I would like to accept questions. I welcome questions mailed at this address. Thank you.